say that you should be more of the system. Maybe
hi everyone. Thank you for coming. It's my pleasure to introduce Benjamin Peter, who is at two places. He's at the University of Rochester and also at the Max-Planck-Institute for Evolutionary Anthropology in Leipzig, in Germany. And I know Ben since he was a young graduate student and I was a postdoc, a young postdoc. And yeah, it's been nice to see your career evolve. And yeah, today he's going to talk to us about his work on Neanderthal ancestry through time. Oh, he did his PhD at UC Berkeley. I forgot to say it. Rasmus Nielsen and Monty Slatkin. And then he did a postdoc with John Bambra. And then he moved to the Max Planck.
Cool. Yes. Well, thanks, Amelia, for inviting me and this nice introduction from when I was still young. And also thank you all for coming. So I know that I always tend to make my talk slightly technical. So if things are unclear, or feel free to interrupt. I think we're still like a small enough group here that, yeah, and I have like, yeah, enough. Yeah, my talk's short enough that you can like interrupt, and I'm very happy to elaborate on things if you're, yeah, because if you don't understand things, chances you're not the only one in the room that will. And so it makes things easier. But anyways, I'm going to talk about some of our works, talking about how we look at Neanderthal ancestry through time. And yeah, as many of you will know, if my slides move, and they do move. Yeah, so modern humans colonized Eurasia, what we think roughly, starting roughly 60,000 years ago. For the exact times, there's a lot of debates and lots of different histories, but sort of, yeah, modern humans originated in Africa, and then over a relatively short time span, moved into Europe and Asia, and then eventually the New World. And when they left Africa, there were already humans there, right, or hominids there, which we call archaic humans. And there's sort of two groups. One are the Neanderthals, which are mainly in Western Eurasia, so in Europe and sort of the western part of Asia. And there's another group called Denisovans that was present in, yeah, what we think mainly East Asia, perhaps South Asia, Southeast Asia. We don't really know. And so when these people, or when modern humans met these people, they interbred, and so now, yeah, and then had offspring, and this offspring contributed to further generations. And so the way we sort of know about this from genetics is about admixture tracks. And this is sort of one of the most important parts of my talk. So I'm going to go through that in some detail. And sort of this is the cartoon that I like to explain this, is that, say, imagine you have a Neanderthal meeting a modern human. So the Neanderthal has two chromosomes, or yeah, two copies of each chromosome. Modern human have two copies of each chromosome. And we know from basic genetics that if they have offspring, then offspring will have one chromosome from, say, the Neanderthal mother and one from the modern human father. So one orange one and one blue one, right? And then if this individual, this offspring, then say lives in a modern human population, what will happen is that these chromosomes will recombine, right? And so the chromosome that this individual passes on will not be the blue chromosome, will not be an orange chromosome, but a mosaic of the two. And if, yeah, so if in a modern human population, it gets another chromosome from another modern human. And if then this individual again has offspring over time, what will happen is that the Neanderthal ancestry fragments in this individual will become shorter and shorter. And so that's what has been used, what we use, and what many other people have also used to sort of trace the legacy of Neanderthals in modern human genomes, right? So yeah, so there's been a lot of work done before, mostly in people living today. And so we know that there's been both gene flow from Neanderthal and from Denisovans in all European and, yeah, in European and Asian populations. And so there's a lot of papers that sort of make maps like this, right, where you have the human karyotype. And in this particular paper, plot here from Vernon Akey, which was one of the first ones, essentially they plotted all the regions where they found Neanderthal ancestry in Europeans in blue and in Asians in red. And you can see that this is all over the genome. There is parts that are inherited from Neanderthals and parts of Denisovans. And so when we started my project, sort of like, yeah, so great, people do this in people living today, but can we also do this in people that lived in the past? So in ancient DNA. So ancient DNA, right, is DNA that we extracted from fossil bones and teeth, and it comes with some challenges. So one is that there's often not that much DNA left, right? And so what DNA there is has relatively poor quality. And so what we have to deal with is, often have to deal that we have only low coverage, so we cannot sequence high quality genomes, but we only have to attend on sort of what we are often limited by how much DNA is preserved in this specimen. Very often, we also have to deal with contamination, so that, because many archaeological specimens were handled by many people, sometimes over long time periods, they not only contain DNA from the original, where the source of the fossil, but also from other people that handled this over time. And then there's other issues like that the DNA gets damaged over time, which makes it harder to work with, and also that we know relatively little about the population history of archaic and modern humans. And all this makes it not as straightforward to look for these admixture tracts in ancient individuals. And so at the start of this project was essentially try to come up with a method that allows us to do that. And sort of one of the the main approximations that we make is that instead of trying to identify these two chromosomes independently, what we do is actually just ask, how many copies of the orange alleles do you have? So instead of having a diploid sort of inference, what we do is we just say like we either have 0, 1, or 2 copies of the orange one. And so I call this model admixfrog because it, first admixture tracts. And the idea is that, to formalize sort of the inference problem that we try to solve, we have one low coverage, low quality target genome. So this is an individual of interest. And then we compare that to what we call source genomes. These are high quality, say, could be Neanderthals, could be modern humans. The model is quite general in that. That is a way to compare and then try to infer which parts of the target individual genome compares to the sources. And we assume that you have some non-contamination panel. So this is for, since we're in data science institute, I figure I can get a little bit more into detail of the method. So the model is a graphical model based on an HMM where we have sort of 3 layers. So we have the overall, what we like to know is the local ancestry. So whether each part of the genome is inherited from a human, Neanderthal, or a mixture of those. Then given the local ancestry, we have one latent variable, that's the genotype that we integrate over. And then given the genotype, we also model the sequencing data, what we actually can observe by sequencing ancient hominins. And we also model that. And so to give you a little bit more detail through all these layers, for the sort of the bottom layer, for the local ancestry, again, what we have is sort of these 2 copies of Denisovan and Neanderthal ancestry over the genome. And so what we do there is that we, it's a little bit dark here, but maybe if you were lucky and got a seat in the front row, you can still do that. So what I do is try to abbreviate that and saying that here, for example, we have Denisovan ancestry, or a diploid Denisovan ancestry. We would have, say, diploid Neanderthal ancestry, and then usually I just use the concatenation of Nia, and then for the heterozygous ancestry, where you have one haplotype from a Neanderthal and the other one from a Denisovan, right? And so we do that in small parts of the genome, usually about 0.002 centimorgans, which is about 2,000 base pairs, which makes it easy to estimate all the parameters directly from the data. Then given those ancestries, so the idea is that if we knew that a particular region was inherited from, say, a Neanderthal, we can then, and we assume sort of how Neanderthals, what genotypes you have, we can sort of ask, calculate the probability of a particular genotype at any particular site, given that it has Neanderthal ancestry, right? And so the way we do that is what's called, so we do it for each of the sort of possible local ancestries, and we ask, we ask what's the probability to say have a derived allele at any particular locus using what's called the Baldwin-Nichols model, which can incorporate sort of the main sources that of uncertainty that we worry about. And so to get a little bit more into detail here, so the way we think about this is that we have our target genome where we have two chromosomes because it's diploid. And then we have our source where we have some individuals with ancestral alleles and some with derived alleles. And so what we worry about is that either the two copies we have in the target come from the same introgressed ancient genomes, in which case they have coalesced or they have a common ancestor before they were introgressed, or there might be two independent chromosomes that introgress separately. And to sort of take that into account, it's sort of depending on how long this time here is from the target to the introgression time, this probability of coalescence will vary. And the second thing is depending in the source population, like depending on how closely what sequence source that we have is to the source that introgressed into the population, there may be more or less allele frequency change in this population. And this is sort of the second source of uncertainty, right? So if you have a Neanderthal that we can compare to that's very close to the one that admixed with modern humans, then this uncertainty would be very small. But if you had only a Neanderthal that's very distantly related to the ones that actually contributed to humans, then you would have to take it into account. And then the last source of uncertainty is that we only have like a small sort of subsample of the source population allele frequency. So we don't actually know what the allele frequency is, but we have to estimate that as well from a usually small sample. And yeah, I'm not going to go through the math, but the nice thing is that if you do this model and make some simplified assumptions, you can essentially get into you can get the genotype probabilities for under this model in using this analytical forms, which means it's very fast to compute and very efficient to estimate all the parameters from the model. Then the last part, and this is again somewhat technical, so the ones of you that are not worried about ancient DNA might sort of zoom out for the next 2 minutes. But so essentially one of the issues is that contamination is for us by far the biggest issue in terms of getting accurate data, right? And so what we wanted to do is to model contamination as what's called the genotype likelihood. So we don't say we know the genotypes, but we assume that we want to model the read data, the sequence data that we see given the genotype. And so what we know, there's some covariates that we know that affect contamination rates. So for example, we know that over time, DNA molecules in ancient DNA become shorter and shorter. And so that means that if you have short molecules, they're more likely to come from the specimen of interest, or our endogenous, as we call them, than longer molecules. So what we can do is we can use the fragment length of the molecules that we recover as a covariate to estimate contamination. We also know that ancient DNA gets damaged over time simply because it's it's sort of somewhere in the bone or bound to some bone minerals or somewhere else, which sort of makes it prone to damage. And so we know that ancient DNA often has sort of a characteristic profile of deamination at the end of the reads. And like as you can say, if a read has deamination, it's more likely to be contaminated than not. And then the last sort of covariate we use is that there's different ways to prepare DNA libraries. For example, you can treat them with bleach or phosphate that have been shown to preferentially destroy contaminant DNA. And so based on this, whether we, for example, know whether a library is treated with bleach or not, we sort of might expect different proportions of contamination than if that's not the case. And so taken together, we can sort of get a probability that a particular sequencing molecule that you have in our data actually comes from, say, the Neanderthal of interest or from contaminant, and in this case, we would want to discard it for analysis. Right. So to give you some sort of to show if this actually works, we sort of did some. We're just going to show you sort of some proof of concept where we use sub-Saharan Africans as sort of a source for modern humans and also as putative contaminants. And that's simply because Africans have the least Denisovan and Neanderthal ancestry among human populations that we know. Then there's 3 high coverage Neanderthals that we can use that have been published as a reference for Neanderthals and 1 Denisovan. And so sort of as a starting point, what we do is we look at genome from a fossil called Ustishim that's been dated to roughly 45,000 years ago. It's modern human. And sort of what we get out of this is again sort of these sort of karyotype plots where we have all the human chromosomes, and you see in blue here regions where we infer this individual to be heterozygous for Neanderthal ancestry, and in orange regions where it's heterozygous for Denisovan ancestry. And you can see that there's quite a lot of blue here, very little orange, meaning that this individual had quite a bit of Neanderthal ancestry, but no apparent Denisovan ancestry. And so because this is a high quality genome, it's roughly 40x coverage. So each position in the genome is covered roughly 40 times. There's other methods that can deal with this data as well. And if we compare those, we see that we get overall very comparable analysis, right? So I mean, Mila told me that Laurit Skoff was here, who worked on that method. And you can see that we run his method on this data, we get very similar results as well. But then where these other methods struggle is that if you have less data. So one thing that we did is then just discarded 99% of the data. So it's instead of a 40x genome, what happens if you have a 0.4x genome? And we find that overall, we can still get very similar estimates. So this is just chromosome 1 here, I have to say. So just for display purposes. But you can see that overall, you find the Neanderthal ancestry almost all in the same regions for pretty much all of them. And we can go down further to 0.1x, and it still works reasonably well. But then if you go to much lower than that, it starts to become, impossible to infer many of the fragments. We can also do other sort of quality controls, ask, instead of using Africans as a reference for humans, what if you use, say, East Asians or Europeans or Africans from a different panel from Southern genomes? And we see that it doesn't really matter. And so this is sort of the method that I'm going to use for the remainder of my talk. I'm going to talk about 2 stories today. And so the first one, I would like to talk about gene flow between. Yeah, sure.
So
That's a great point for asking questions, yes.
you said you're using HMMs, so
Mhm.
you get some probabilities.
Mhm.
Here, you're just giving them a color. You're not
Yes.
showing the probability. So
yes.
are you just sort of taking a majority vote and throwing away the probability information, or will it reappear later?
Yes, it's a good question. Wasn't going to go into detail on this. So what we do, so this is the posterior decoding of the HMM, right? So it's, you can see that, for example, here with a fragment where you say it's 50% probability. So what we actually do is I have a sort of algorithm that calls fragments given the posterior. So essentially what we do is we try to find sort of regions where you have like a consecutive stretch of high probability of Neanderthal ancestry, and then use those as a, as a fragment.
And my other question is at the boundaries where there's
Mhm.
change
Yeah.
from one state to the other state.
No, I do not. And I mean, the reason for that is mainly that a lot of the data we're going to use has not been actually sequenced, but it's been sort of enrichment captured. So we actually often don't have full sequence data, but we just have SNP data. But yeah, it's a very good point. Other questions about the methods? Yeah. Mhm.
The downsampling here is to show that you can correct well for the contamination.
No, so there's no contamination added here. It's just coverage. Yeah. So I think I also did a test where I added contamination, but it sort of looks similar. Yeah.
So
Mhm.
I guess my understanding is like when you go from the ground truth, the genotype, and then to the sequence series, I think,
Mhm.
is it like very much in parallel, or is there any like dependency or like some dependency between like if I know one sample from a Neanderthal, is there any like dependence for the next sample maybe down the read for it to be from a Denisovan ancestry?
So in terms of so the way I do it like independently for each individual.
Okay.
Yes.
So it's like for every bin, it's independent from the next bin over. You're looking at like just that.
Yes,
one
given the
sample.
given the parameter each. So I mean, there's 2 things, right? So if you talk to sample about, if you say sample, do you mean like the sequencing reads, or do you mean individuals?
Oh, I think I meant like for like a given bin, like
Mhm.
is it from a Neanderthal, like the modern bin?
Right. Yes. Yeah. So essentially, the way it works is, right, given the so this is the hidden state here, right? And so this given this yeah, everything genotype and reads is independent given, yeah, the hints that, yeah, I mean, the only dependence comes in through shared parameters, right? For example, yeah, with this contamination model, we have, say, shared contamination parameters that then don't care where exactly on the genome the read is. It just cares about what are the sort of these covariates. Yeah. Other questions? You guys are great. Anyways, let's talk about Neanderthals. Yeah, so I think I very briefly mentioned it, but I just want to give you a little bit more overview about Neanderthals and genomes of Neanderthals that have been sequenced, right? And so this is sort of an overview about Neanderthals that have been sequenced. So there is 3 high coverage genomes. 2 are from southern Siberia, from Czechoslovakia and Denisova cave. And then 1 is from Croatia, from Vindia cave. And then there's a few, about 10-ish or so lower coverage genomes from Europe. And there's also about 10-ish from Czechoslovakia cave that I didn't add here because they're from the same site. And yeah, so this is sort of what about Neanderthal, which doesn't seem that much, but it's a lot if you compare it to Denisovans, where the fossil record is a much more sparse, right? So for Denisova, there's 1 high coverage genome from Denisova cave, and then there's 3 low coverage genomes also from Denisova cave. Again, so it's the same site where also 1 Neanderthal is from, which is why I usually refer to this as the Altai Neanderthal, even though it's like sometimes it might be called Denisova 5, but sort of I try to keep the word Denisova for Denisovans in this talk, and yeah, use other words for the Neanderthals. And there's some evidence from other sites, like from there's fossil data and more proteomics data from this site in Tibet, and there's some other sites in China and in Laos that have been suggested to be Denisovans, but overall the record is very, very sparse, right? And so we know that these groups have met in part because they're both from Denisova cave, but there's also 1 individual, Denisova 11, or people call it Deni, that is an F1 offspring between Neanderthal and Denisovan. So direct evidence that they must have interpreted. But what I wanted to do for this project is to go a little bit deeper in sort of seeing have they met just once or methods have been recurrent. And yeah, to give you a timeline on what when Neanderthals and Denisovans interpret. So this is just samples from Siberia. Like Denisova 2 is the oldest sample that's pretty inaccurate, this dating here. So here, the lines here sort of reflect uncertainty that we have about the dates or ages of this individual. And it's, yeah, sequenced to roughly 0.3x. So about 30% of the genome have some data, but also like 90% of that data is estimated to not come from Denisova 2, but from some modern human. Then there's also Denisova 8. It's a more recent Denisovan, roughly 120,000 years ago. And then there's the high coverage samples that have very little contamination. And then, yeah. So I want to focus at first at this Denisova 2, which is sort of, yeah, sometimes the biggest challenge. And yeah, so one thing that we could do with our admixfrog method is to first look at the contamination that we get. So again, this is just from the part of the model where we calculate the genotype likelihood, right? So probably of the genotypes given the reads. And so what I did here is call it just the proportion of the data that we estimate to be deaminate or a contaminant and non-contaminant. And I split it up for reads that have a deamination or ancient DNA damage at one end and reads that don't, right? And see that most ancient DNA analysis do in the present day is essentially, if there's evidence of contamination, they just focus on this part here and toss out this part, right? But you can see that for this particular specimen, this will not work very well. For once, there's still about 20% contamination left if you look at the deaminated part. And then the other part is you do actually toss out about half of what we estimate to be the endogenous data for this individual. So really showing that modeling contamination in this case here can sort of actually help us work with a specimen that otherwise would not be analyzed easily. And then if you run the HMM, what we find in the posterior is that we do in fact find fragments like here or here, where we do find evidence of fairly long tracts of Neanderthal ancestry. Right? So here I have Denisovan ancestry in orange, Neanderthal ancestry homozygous in blue. and then the heterozygous ancestry is in brown. And you can see that there's some fragments here that are brown and fairly long, and yeah, so they might look short on the screen, but they're like several million base pairs long, right? And yeah, so one thing you can ask, when I the first time saw this, I asked, is this actually real? Can we validate this somehow? That what this somewhat complicated model spits us out to make sense. And what I came up with as sort of as a validation analysis was just look at the data a little bit more carefully. And so the way I do that here is sort of look at one of the regions. I look at this sort of section here, which is the longest region of, yeah, inferred Neanderthal ancestry in this genome, right? And sort of ask what are the different, actually the raw underlying genotypes, right? And so what I have here is sort of pick out all the sites where the Africans or all Africans that we have in sort of a reference panel have one allele that is sort of set at the frequency 0, and then either Neanderthal or not Denisovans, but not both have a different allele, right? So in this case here, for example, this particular site here, all the Neanderthals have one allele, sort of is at the frequency 1, the Denisovan and African have the other allele. So in this case here, right, so if our target individual had a Neanderthal allele here, we could be fairly confident that this is from a Neanderthal ancestry tract and not from contamination, right? Again, because in this specimen, we estimate about 90% of the data comes from contamination. So we want to make sure that we don't look at contamination or validation analysis. And so when you look at our individual, Denisova 2, what we find that in this region here, roughly half of the regions are blue, or SNPs sort of we match the blue sites and half match the orange side, right? Which is exactly what you would expect if this individual indeed, or this particular part of the genome was indeed heterozygous for Neanderthal and Denisovan ancestry. And sort of in the faded on the sides, you can see that outside this region, it looks like it's all orange, meaning all Denisovan ancestry. Again, what you would expect if this individual was a Denisovan, right? So it's sort of, I think one way that helps me sort of get some confidence that the result that we get here is actually a real signal and not just some artifact of our method. Right, we did then the same thing with Denisova 8, which is another Denisovan that's dated to roughly 120,000 years old. And again, it's sort of a similar pattern. So Denisova 2 has a bit more data, right? And so you see that the patterns are overall quite a bit clearer. So we get sort of sharper boundaries. But again, we see that fairly long fragments up to 10,000 base pairs, or sorry, 10 million base pairs long of Neanderthal heterozygous ancestry. We also see on the X chromosome, the one fragment where this is all blue, and this is because Denisova 8 is a male, right? So it's only haploid, so it has only one copy of the X chromosome, and so we call this as a haploid Neanderthal intergressed fragment. You can also do the other question, ask, do the Neanderthals, like the Alpine Neanderthal from the Denisova cave, does it have any Denisovan ancestry? And again, the answer here seems to be yes, in that we find several regions that are about 1 centimorgan, or roughly 1 million base pair long, that do come from a Denisovan in this case. Again, the color scheme is the same, but now because this is a Neanderthal, it's mostly blue, and there's no orange fragments because there's no homozygous Denisovan ancestry fragments. But then if you look at later Neanderthals, either from Siberia or from so these are all Neanderthals from Europe, we don't find any evidence anymore for Denisovan ancestry. So as a summary, it seems like Neanderthals, so essentially we find in these early Denisovans that they had a lot of interactions with Neanderthals. Yeah, sort of just looking at where the fragments are on the genome, it doesn't seem like it's the same event. It seems like this individual had different events of gene flow. And then also in the oldest Neanderthal that we have, we do find some evidence of the Denisovan ancestry, whereas in the later events, or later on, this ancestry seemed to have disappeared. And sort of what we think about is what might have happened is that so one way sort of we can make sense of this is that if Neanderthals were mostly in Europe, and Denisovans were mostly somewhere in maybe East Asia or Southeast Asia, and neither of them was like particularly often in South Siberia, but there sort of was one contact zone where they went to, but then there was relatively little gene flow or movement back from Siberia, back to, at least back to Europe, right? Because if there was, we would expect to see Denisovan ancestry all through the European Neanderthals, but we don't see that. So it seems like, yeah, what happened in Siberia stayed in Siberia, and the Denisovan and the Denisovan ancestry does not seem to have made it out of there, at least not in long term. So this is sort of first story I want to talk about. The second story is work with, it's a collaboration with Priya Morchani and sort of my student Leonardo Yassi and Priya's former postdoc Manshusha have been sort of driving the sort of legacy of Neanderthal ancestry in modern humans. Right. So what we wanted to do is ask, so I mean, the funny thing is that Avi here had to present this paper in like quite some detail in the was it a training grant seminar? So I have to now present it back to you. Anyway, so what we were trying to do is to sort of try to systematically map how Neanderthal ancestry got distributed in human genomes and sort of learn more about when this gene flow happened and why it got spread out like this. And yeah, so this is sort of an overview of the samples that we used. So we used 59 ancient samples and about 300, almost 300 present day people. And sort of this is sort of, just sort of a map and sort of a timeline of the sample that we use. And if you know engineering, you know that there's now tens of thousands of samples. But what we really wanted to sort of focus on this period before the last glacial maximum, which is closest to when the gene flow happened. And for that time period, the data is still very sparse. So that's why we use this relatively small sample set, because we sort of really wanted to focus on this early time period. No, so some are captured, some are sequenced. Yes. Yes, that's a good question. Yeah. Mm-hmm.
You did the primary estimation. I assume that you're using like a Olympus model.
Not necessarily. Yeah. Yeah, not necessarily. Yeah, I mean, maybe yes, it's somewhat implicitly, possibly.
I guess my understanding was in that model, including your emissions model, you have essentially the timing parameter.
Yeah.
I was just wondering if when you do parameter estimation, if that timing parameter is actually informative of essentially the timing of gene flow, and if like there is information just in the HMM alone without going to this paper that I know you're about to present on for inferring these times.
Potentially, yes. I mean, I think it's so I think there's a couple things I want to mention here. So one is that the model that I'm talking, or the model is very implicit here, right? So essentially, the way I think about time, and probably should not go back to the slide, but essentially the way I think about time is sort of in what's the probability of coalescence, about probably that these two lineages have coalesced or not, and that is closely related to time, but it also depends on, say, population history and other factors we don't want to explicitly model. So the way that's why we sort of do actually the dating in other ways. I mean, you sort of, and so I mean the other part is that you're right that the model assumes a time, but in some sense you can think of this as a prior, right? And then the actual fragments that we get are in some sense, I think, more informative about timing than the inference parameters that we use. Other questions here? Anyway, so just qualitatively what we get is sort of, so similar of what we showed, right? So this is a person that lives today from Hungary. And you can see that the Neanderthal ancestry fragments we get in this individual here are,
Going back to the fact that some of the ancient genomes are the same for race, I just like looked at. Could you say something about the Arctic admixture rate?
So that's the one that we use for this. Yes.
Could you just say something about it? Like how's the distribution of this?
So this is essentially sites where people looked at the Altai Neanderthal, so this one from Denisova cave, and one African genome, and looked at sites where these two individuals differ from each other.
But like with that array, like what's the I mean like the distribution of the sites?
Yeah, it's about
between the
1.7 million sites across the genome.
Like
It's quite a lot, yeah.
Okay, just like I was just like curious because like I mean I can like tell you later because it's like
Yeah.
person, like,
Yeah. Maybe that's it's sort of like a very somewhat technical question. I think maybe
Yeah, sorry.
that's okay. That's okay. I mean, I think it's always great if students ask questions, so I think it's great. Anyway, so yeah, so for a present day people, we get like these very short fragments, but then you can I think already see visually, right, that if you look at the individual that lived 30,000 years ago, the fragments are a little bit longer. And you look at an individual that lived 45,000 years ago, the fragments are much longer in comparison. Right. And so one thing that we wanted to do, the first question was like, was this one event or was this multiple events? And so to come back from the cartoon from the introduction, right? So the way we think about this is that, so we can have sort of the Neanderthal and modern human sort of mix, right? And so F1 again has one chromosome, then F2 would have sort of these recombining chromosome. But then over time, right, there would be multiple individuals that all have this Neanderthal ancestry. And sort of the idea is then if these two populations or this individual have ancestry from the same Neanderthal integration events, then the fragments that they have might overlap in the in where they are in the genome, right? Because in this example here, they would all might have only Neanderthal ancestry in this particular part of the genome, but they might not have Neanderthal ancestry in other parts of the genome, where this earlier individuals did not have Neanderthal ancestry. Whereas in contrast, if we had sort of a second Neanderthal that integressed, and here in sort of one of the first generations, we had Neanderthal ancestry in different parts of the genome, then you would expect the Neanderthal ancestry in sort of one individual that had ancestry from here, another one that had ancestry from here to be largely uncorrelated in where they are in the genome. So the first thing that we did was just do a principal component analysis of the Neanderthal ancestry locations in the genome. So we just asked, given where you have Neanderthal ancestry in the genome, are these patterns clustered or are they just random? And so the colors here are geographic regions, and you can probably already see that this sort of, that the same color plot or same colored individuals do cluster with each other and are close to each other. And it sort of reflects geography, right? For example, these are, I believe, Western Eurasian individuals. These are Siberian, East Asian individuals. So we have sort of this east-west gradient in the, in terms of the diversity and sort of this, Then the question is like, is this random? Is this sort of unexpected? And in some sense, what we do is then we took these sharing patterns and just did the plot here on below the diagonal, sort of the correlation that we have in the sharing patterns between individuals that sort of the PCA basis on, and then above the diagonal, what we plot is just a measure that's just based on the genome-wide average. So this doesn't care about Neanderthal ancestry at all. It just asks genome-wide, are you similar or not? And you can hopefully appreciate that the below diagonal and above and below diagonal parts of this figure overall look very, very similar, right? That they seem to be overall, correlated, sort of indicating, so you can also quantify this and show that, we get a very high correlation coefficient between the two pairwise comparisons, sort of indicating that overall that sharing pattern of Neanderthal ancestry can be largely explained by sharing of just shared genetic drift or shared population history. The one exception to this, and this is a little bit hard to see, are sort of these four individuals here, which are one of the earliest, which are some of the earliest samples outside of Africa, which do not seem to have any sharing with other individuals in terms of Neanderthal ancestry. Then sort of a second analysis we did is looking for unique fragments. So we ask, are there particular parts of the genome where we find Neanderthal ancestry only in one particular population or one particular set of samples, but not in others. And yeah, so what we find here is that in the earliest samples, so what we call the early out of Africa samples over here, is that we find a lot of unique fragments, or a lot of the genome is in unique fragments, versus in everything else, so all the other, yes, here's just a, yeah, again, a sample overview with the same colors, seem to have overall very little unique entries, sort of indicating again that perhaps these earliest samples had some different Neanderthal gene flow, and then after that, everything sort of became normalized and is mainly due to shared population history. Then, yeah, sort of the second analysis is sort of how we want to, yeah, so back to Dave's point, and how you would like to date Neanderthal admixture. And so, again, these fragment lengths here are sort of have some implicit information about the locations of where, yeah, or how long ago, how the length of fragments have information about how long ago gene flow has happened. Now, because of technical reasons, because, for example, hard to estimate the boundaries of fragments, we actually did not use the fragments themselves to do this dating, but we actually used a covariance-based method that Priya has developed to sort of get sort of a, yeah, on how much the covariance or something very closely related to LD decays over time to try to date our method. And so for this covariance measure, you can see for old samples or that have Neanderthal ancestry in the recent past, you have this very slow decay of this measure over time, versus for more recent samples, like the 7,000-year-old samples, you have this very rapid decay over genetic distance of this, yeah, this measure. So as sort of a mathematical aside, this is actually, these two measures have the same information. So meaning that if you have these, yeah, if you have this, the length distribution of fragments, you can derive this decay curve. And the same thing, if you have this decay curve, you can derive the length distribution of fragments. So there's this one-to-one relationship between these two. Yeah.
I think Neanderthals are essentially like diploid ancestry. Like, so then necessarily like having a heterozygous segment of length L is not the same as having like a segment.
Yeah. So the way I think for this paper we just ignored this because the proportion of the overall ancestry proportions are roughly on the order of 1 to 2%. Right. So you could sort of make an assumption that, yeah, you could sort of try to, like, if you assume the chromosomes were independent, you could kind of just say you have independent fragments, in which case you get again an exponential distribution, but with like a 1 minus m factor added to it. So we're off by a factor of 1 to 2% by ignoring that. Yes. Okay. So yeah, so one thing we want to do is essentially ask, so one thing we want to do is ask not only like when did gene flow happen, but how, over which time period did gene flow happen. And so what we use is what we call this extended pulse model. So where we say that we estimate both a sort of location parameter when the gene flow happened, and we estimate a duration parameter when, however much time period gene flow happened. And so using this allows us to use just two parameters, one for location, one for the duration, to give us some more information about when gene flow happened. And so what we find using this is that we estimate, and that, so overall we find that the data fits rather well, or relatively well, to the model, and we get sort of a mean time of gene flow of roughly 47,000 years ago, and a duration of roughly 7,000 years. So there should be a K here somewhere. Yeah. So again, so there's sort of some samples, like the most recent samples. Yeah. So this is sort of one way. Again, we could sort of think about this. Is sort of this is what we think when gene flow happened over which duration, and these are all the sort of different individuals that we used for this dating. I think, yeah. Then sort of should start to wrap up. But so we wanted to talk a little bit about what we learned about the biology of Neanderthal ancestry and how it impacted us in this is sort of the first thing we did is just sort of map where in the genome do we find Neanderthal or non-Neanderthal ancestry. And so here in orange, you have all the regions where we do find Neanderthal ancestry, and then blue is regions where we only find modern human ancestry. And then in green here are regions where people already in previous studies found that there's a depletion of Neanderthal ancestry, and you can see that we find those as well. So these regions that have been found in present day people were also already present in ancient people. Yeah, so there's a few individuals that seem to have some fragments in those. So these are sort of just to zoom in on these regions of depleted Neanderthal ancestry that we call Neanderthal deserts. That yeah, there's a few sort of fragments that sort of are close to the boundaries or in this fragment, but overall, these seem to hold even in the ancient data. Then, so one thing we wanted to do is then look at, are there regions that change in frequency over time? So are there regions that were either, so here's the frequency in ancient individuals above the X, or yeah, up here is the frequency in ancient individuals, and here's the frequency on present day people below the X-axis. And sort of we looked at sort of 3 different patterns. Like one is regions like this one here, where we have sort of a high frequency in both ancient and present day people. Then we have regions like this one here, where present day people have high frequency, but ancient individuals had only low frequency. And then the opposite, right, where ancient people had high frequency, but then present day the frequency has decreased. And so we find regions of all of these patterns, yeah, in the genome. So for example, one region, the one in blue, we have 3 out of, I think, 22 individual, or no, 3 out of 18 individuals have in the ancient pattern have high frequency, and then sort of but then present day has yeah, has already is the right example? Yeah, so this is I think a region that's been always at high frequency, or at least versus, for example, this region here. And so yeah, so one way you could say about it that here selection must have happened relatively rapidly, right? So the reason that so 3 out of 18 might not seem a lot, but if the genome-wide average is between 1% and 2%, right, this is still like a significantly higher, right? But then we also have regions like this one here, where we find only essentially one individual in all our ancient data had this individual, but then roughly 20% in the present day have this Neanderthal haplotype, which sort of might imply that if there was selection, it happened not immediately after gene flow, but much later in the more recent history, right? And then if you sort of find the opposite, like in the red region, what we think might have happened is that this was either immediately beneficial, perhaps because it's like yeah, a region like this might have sort of had some resistance to pathogens or something that was like a sort of a transient selection pressure, but then sort of got depleted again. Or it might also have been just close by to a selected site and then recombined off and got reduced in frequency. Then the last thing I think I want to talk about here is that if you look at the X chromosome, then the pattern looks quite different. That the X chromosome, we have one much less Neanderthal ancestry compared to the autosomes. And sort of the other pattern that we see is that it is much more clustered in like a few select regions than yeah, on the autosomes. We don't quite know why. We know there's been sort of a lot of sort of recurrent selective sweeps on the X chromosome. So it could just be that, but it could also be that there's partly some incompatibilities that may have contributed to this. Anyway, so in summary, I'm going to wrap up. We have presented for this project a catalog of Neanderthal ancestry in ancient modern humans, sort of a time series of Neanderthal ancestry in many different individuals across the world, or at least across Eurasia. Over time, we think overall that all the Neanderthal ancestry that we have in present-day people comes from one single event or extended event for over 7,000 years, and that there was selection that probably happened relatively early on for the most part. Anyway, so this is all I wanted to tell you. I also like recruiting, and yeah, many thanks to the folks in my team and you all for coming to my talk. Yeah.
It's a really exciting talk.
I have a couple questions about admixfrog.
Mhm.
So I'm curious if you, when you were developing a method, if you tested specifically, I guess, like its accuracy or sensitivity when it comes to calling, I guess those heterozygous tracks.
Mhm.
Just because I guess depending on how, like if a track was truly heterozygous and how they were treated, there could be varying degrees of overlap. And so I was wondering, like, if you understood some dynamics of like a minimum proportion, I guess like of like how much of a track needed to be within your 0.02 centimorgan bin needed to like be able to confidently call those heterozygous versus homozygous tracks. And my second question is that if there's any information either within an individual and amongst all their reads, which I doubt there are, but if you think there is like a follow-up for if you incorporate multiple reads to then essentially kind of maybe try to phase the specific heterozygous tracks themselves by looking at multiple ancient individuals and essentially being able to like conglomerate and essentially have like this cool inequality bases.
Yeah. Okay. So for the first question about, so the thing is the regions that we look at are very short, right? So on the order of like usually 2,000 or 5,000-ish base pairs. So we think like the average length of fragment is on the order of 50 to several 100,000 base pairs. And so I haven't really looked at in much detail what happens within a window, right? If part of it is in a fragment and part of it is not. Just because, yeah, it's sort of like, I'm happy with an accurate, like, yeah, I'm happy to say that the fragments we call have like an accuracy of like several thousand base pairs.
I guess like I guess because you also apply it to modern humans, which the data is better. I guess like when you're looking at windows that small, can you get a heterozygous fall because a lot, I guess, like of the different genotypes are actually heterozygous, or do you have like part of that small window that has, I guess?
Well, it would only be the first one, right? So essentially most fragments you have are like several like tens or dozens of consecutive fragments, right? So if you only have partially Neanderthal ancestry, it will probably only matter for the very first of the fragments. But I don't think, yeah, I hadn't really thought about a scenario, you know, where you had like, say, lots of very, very short fragments of Neanderthal ancestry interspersed by lots of very, very short fragments that are homozygous for modern human ancestry. I think, yeah, it might not be distinguishable, but I think I can't think of a scenario that would give rise to such, yeah, of such data. And yeah. But yeah, in some sense, that's the approximation we make, right? Sort of make this work on low coverage data. Like, I agree, it'd be nice to do it on face data, but we don't want to do it. So what was the second question again?
Yeah, just if you thought about like using information across, I guess, the good reads
Yeah, we thought about it. We haven't really done anything really about that. But I think, yeah, I mean, one could imagine. We thought about it, but it's sort of not quite clear what we would get from that.
I also have a question for you there today. Like a short thing of the windows. So you were saying that for your SNP chip data, we're using this archaic admixture. And so from what I understood that you said before is that the sites that you are like capturing are sites that you like know based on the alpha in your dental that might be in progress. Okay. So if you have like an array that is now this archaic admixture array, it's like, I don't know, mega, for example. And there's something also another SNP chip array called mega, which people usually use to capture admixed populations, for example. And it's just basically enriched for common SNPs.
Yeah.
So do you think admixfrog will work on that? Because you will have like, it is not the same to have.
Yeah, no, it will work a lot less well. Like, so, I mean, there's like, yeah, so I haven't really gone into that because it's like a little bit too technical for this talk, but there is also like most ancient DNA is not actually captured for this archaic admixture array, which is sort of useful for looking at Neanderthal and Denisovan ancestry, but for what this people call like the 1240K array, which is that called because it has 1.2 million sites. And that one, yeah, is sort of ascertained similar to, say, mega array or other like things that people use for Chivas and other things analysis that are sort of focused on human variation. And using that, we have a lot lower power, which is sort of partially why we sort of, yeah, did not use some that were just genotyped on this. You could, I mean, you could impute it, right? I mean, I think if you wanted to go the imputation route, you could do that. I'm a little bit worried about imputation is very early modern humans, right? Because not like there's some evidence, at least some of them have ancestry that's no longer not present in present day people, which is maybe by imputation might be tricky, but I think for many individuals, that's the most practical thing to do. Yeah.
Do you model the recombination rate that is known among cross chromosomes in this? Or?
Yeah, so the way I do it, I use a recombination map as of an input, and then, yeah, based on a human recombination map. I did like try various different recombination maps. So for most of this, I use one that's based on African Americans, simply because, yeah, I thought like these are two somewhat distinct populations that came together. So maybe it's somewhat reflective of human history over the last 40,000 years.
So do those in any way explain or correspond with the deserts versus the non-desert region?
Not specific. I mean, so Neanderthal ancestry is correlated with recombination rate, right? So there is more Neanderthal ancestry in but high, yes, I think where it's high. I always have to think about which way around it goes. But of course the way where you would expect it that, yeah, like if you have some purifying selection.
Trying to separate recombination opportunity versus selection for against the archaic piece, because the local recombination environment will hinder or enable things that are positive or negative to respond.
Right, right.
Can you disentangle that?
Yeah, not for a specific fragment, but yeah, we do find this association where the Neanderthal ancestry is depleted in higher recombination regions, sort of might indicate it's sort of better at escaping purifying selection. Yes.
So going back to Denisovan ancestry in the Neanderthals. Did you find Denisovan ancestry? Which ones did you find Denisovan ancestry? Did you find it in Cheshire prior to?
Not to a meaningful amount. Very little there. Like, I mean, sort of like really short fragments in the sort of point that I don't really trust them. Because they're short enough that it could be something weird going on.
But I guess the Denisovan is
yeah, I mean, the Denisovan is much younger actually than this. Like the reference Denisovan that we have is much younger than this individual. But yeah, it's just like the fragments are just sort of on borderline on where we sort of think that we have high confidence to call them. And yeah, then we come back to the recombination rate that if you knew the recombination rate Neanderthals, I would be somewhat more confident, but it's possible because we don't really know the recombination rate in Neanderthals, that these are just regions where, yeah, we might underestimate the recombination rate and the fragments appear longer than they are. Versus for the regions where we have like, yeah, that are like on the order of a centimorg in a human recombination map, and like a million-ish base pairs on a physical map, those I think are harder to argue that you have this really low recombination rate.
Are those the centromeres, the gaps?
No, so the gaps here are just, so this is I just cut out the centimeters. So these are just the chromosomes. So this is just like plotted here. This is chromosome 1 goes to here, and this is chromosome 22. Yeah, sorry, it's just like, yeah. So just use, yeah, so it's actually not the physical map. So I just cut out the centromeres because they don't really, appear on the genetic map here.
This is a kind of speculative question. To what extent are people looking for more samples, and do you have a kind of wish list, age, location in Eurasia, where
everywhere?
you get more samples?
Yes, everywhere. I mean, really the regions where Yeah, yeah. Yeah, I mean, we really, with the we're really limited by the samples, right? And so my collaborators in Leipzig, they have spent the last 20 years or so trying to optimize the protocols, getting DNA out of Neanderthals and Denisovans.
Okay, but that's for getting the DNA. What about for just finding samples in the ground?
Well, so the problem is that I mean, the thing is, in most other places, there is just no evidence of DNA preservation. Right. So I mean, there's some regions where the issue is sample access, but mostly the issue is DNA preservation.
Maybe I'm not, so
Yeah.
you have some dots here. Are
Mhm.
these all caves?
Yeah, these are various cave sites that have been, yeah, I mean, so in general, right, so there's not too many Neanderthal fossils, period. There's like, yeah, maybe a couple hundred, maybe 1,000 Neanderthal fossils that have been excavated in the history of science.
And so the idea is there are not too many more to be found. And
I mean, it's
therefore
just very
you have
costly to find them, right? I mean, it's just like needle in a haystack kind of question, right? And so people, I mean, there are archaeologists looking for Neanderthal fossils, right? And we know lots of sites where there is Neanderthals that have no fossils, right? So one thing that people do that I think is very exciting for this work is they actually find Neanderthal DNA in sediment data. So that just from dirt at Neanderthal sites, they find, yeah, they get DNA out of that. And I think this is really something that can help here. And so I and many other people sort of working on, related methods that just work with really low, even less amount of DNA. We just have, say, hundreds of molecules that we can assign to a Neanderthal. And so sort of try to make some meaningful inference from that. So this is sort of an avenue that's happening.
Is there some biochemical characteristic of the soil where it's better to?
Yeah, I mean, people are sort of looking into that, looking more systematic. I mean, we know in general that particularly temperature and humidity seem to be the major destroyers of DNA over time. For soil, I think it's less clear. There's no less obvious patterns. Yeah, so some of my colleagues in Leipzig are working on sort of more overview studies like that. So yeah, in general, it's sort of the entire field would like to have more samples, but it's really yes, they're not easy to get by.
Thanks, again.
Yeah, thanks so.
Yeah, right. How you doing?
How are you?
Good, good.
Yes, yeah, we've got microphones now.